Hello, good morning, everyone. Welcome to the fourth lecture of our Human Practices series for Synbio 1. We're going to be talking about governance, specifically about how synthetic biology is governed and how the different approaches are to regulating synthetic biology. So we'll talk about the forms of governance, the basic principles that are currently being used. Specifically, we'll dig upon a little bit more on responsible innovation, general approaches mostly by states and nations for governance, what the current state of synthetic biology regulations is in different major countries, as well as a brief look into risk assessment and how that's been applied to synthetic biology technologies. So, first of all, let's define what governance is. Governance is generally just the oversight and regulation of any activity by an actor or a group of actors. This actor can be anything from the government to the scientists to funders, and we'll see more later. Really, there's no one rule, one-size-fits-all model for governance. It very much depends on the technology and the application being involved. For example, something that is to be used for remediation and to be released into the environment, like a bacteria that, say, remediates plastic or any kind of toxic substance, would have different concerns than, say, something that is just used as a diagnostic test. You also have different moral concerns, be it depending on what it is being applied on and who is involved. You have different stakeholders, different people, different consumers, different people involved in the industry, as well as there are different background constraints. Intellectual property is different across different industries, and there's different regulatory frameworks that apply depending on what product or what technology you're using. So for example, something that has medical applications would be regulated differently than something that is used, say, in a manufacturing setting for something that is not to be used for, doesn't have any therapeutic effects. Let's just look generally at the forms of governance. I drew a lot of these forms from a course on ethics and bioethics and synthetic biology by John Hopkins, which I'll talk about some more at the end of the video. But generally, the norms of governance are generally just standards of behavior. Thus, they are generally voluntary and applied for by people in a specific field. So, for example, by scientists or by a particular commercial industry. They're a set of rules that essentially everyone agrees to work upon. The examples in synthetic biology, the very first ones was by someone called Andrew Lewis, and essentially he had developed a hybrid form of adenoviruses that could potentially be able to integrate foreign DNA into it and could have the possibility of transfecting that into eukaryotic cells. At the time, that was a very novel technology, and people were concerned about it being used to cause different types of cancers in animals and humans. So Andrew Lewis had himself created norms. Like he had a very stringent vetting process with regards to whom he shared his technology with and his research with. The SB 2.0 resolutions are another example of norms, which are basically the SB 2.0 was a conference after which that involved scientists and many people outside of science as well, after which they came up with a bunch of resolutions on what the standards for screening DNA synthesis material or information on the customers to which these oligos and biobricks would be distributed to, as well as general resolutions on biosafety and biosecurity issues. So what norms are generally are just things that the industrial scientists agree upon doing. There's no actual legally binding framework to them. You have self-governance, which is again self-imposed rules by individuals and professionals and industries. These are kind of more widespread across an entire industry. In Synbio specifically, DNA synthesis companies have over time developed standards for the industry with regards to what sequences they are willing to synthesize for people, given the risk of it. Um, and as well as having some kind of tools in order to screen those sequences that they will synthesize, in order to make sure that they're not creating anything that could be used by people for or misused by people in some way. The Asilomar conference is an example of a conference where governance, that's those self-governance rules were actually discussed. So just to give you a bit of a context, because it's a pretty big deal in Synbio, the Asilomar conference was happened in 1975, following another conference where they had found for the first time that they could actually add foreign DNA into bacteria, and that they could, using viruses, and then that the applications that that could have with regards to potentially providing some pathogenic or more infective properties to recombinant organisms. And after that first conference, they had a moratorium saying we're not going to do any kind of research of this sort until we've properly discussed the ethics of it. And that's what led to the Asilomar conference, where scientists and various other stakeholders and ethicists were brought together to discuss what the rules of the industry, what the guidance would be for how recombinant technology were to be used going forward. And they had a lot of guidance on, the physical containment or the biological containment that needs to be set for a certain technology to be used. So say you're using a more potentially virulent or contagious vector or DNA strand, you may need more a higher level of biosafety level in order to contain it, or in terms of biological containment, in terms of making certain potentially pathogenic model organisms non-viable outside of a laboratory setting. So governance is also instituted by institutions nowadays. Universities and research institutes have various things like material transfer agreements, IP and licensing agreements that govern how new discoveries, how material comes in first into labs, and also how new discoveries are distributed into society and how they're legislated in terms of the intellectual property and the rights that go with it. We also have institutional biosafety committees that vet research and and determines whether there are any ethical or safety concerns with them before they're allowed to move forward. Regulation comes also at the level of funders, and you'll see as we go along through these examples, very much the governance comes from various stakeholders, and the influence and how they govern it depends on what the relationship of the stakeholders are to each other and to scientists in the field. So in the case of funders, the relationship is that of research funding and how the money is used in research and commercialization. So this can be both public or private, so say industry funding or the government NIH funding. And these institutions have provided various regulations on how the research can be funded and how it can be licensed. So, for example, the NIH has a clause on gain of function research. Basically, gain of function research is research on strains that can lead to enhanced pathogenicity or transmissibility of potential pandemic pathogens. And there's obvious biosafety and biosecurity concerns with those with regards to them being used for something we call dual use risk, which is basically being used or misused in a way that could lead to things like bioterrorism or just being able to spread pathogens that could be dangerous to the public. So the NIH has guidelines on how it regulates research that may involve pathogens or synthetic or recombinant DNA of that sort. And as well as with regards to funding, the RIC, previously was called the RIC, the Regulatory Recombinant DNA Advisory Committee, that they used to regulate the research on recombinant DNA and provide essentially the green light for research to be done on different kind of recombinant work. But nowadays that's been shifted to the FDA. They're today more concerned with kind of public discourse and developing guidelines, but from a historic point of view, they've done a lot of the vetting and the regulations with regards to how what research is considered adequate for funding. It also happens at the levels of journals. So, as most of you know, a lot of synthetic biology research is at the academic level, and most researchers want to have their research published. And journals are therefore at an important position in able to regulate what kind of research actually becomes published through their publication, and that has implications on, they're essentially gatekeepers of what research becomes available to the public. So they've had various statements made by different journals on how they deal with dual use research. So again, dual use research or research for which if the information could be misused for potentially dangerous uses, and data sharing. So with regards to transparency and how data is being shared to everyone, or with regards to whether if some information is sensitive or potentially dangerous, how they're going about taking that out of widely available published information in papers. So you also have regulation or governance at the level of states and nations. So that can be direct governance in terms of laws and regulations, but it can also be at the level of funding, as we discussed earlier, in terms of what research receives governmental funding. The US, for example, with regards to laws and regulations, has rules on the use and distribution of human tissue, stem cells, and human embryo, and what kind of research they consider adequate or ethically within the realms of what that specific nation will allow. There was also an important event in synthetic biology which was involved the Dutch government. Essentially, there was a doctor, Dr. Fouchier, who essentially developed an H5N1 flu strain that could infect ferrets via airborne transmission with only a few mutations, and he wanted to publish his research in the Journal of Science. And essentially, the National Science Advisory Board for Biosafety in the States recommended that certain information be taken out because obviously research on mutating the H5N1 to be potentially more increasing its transmissibility could be something that has biosecurity concerns. And while the Science Advisory Board eventually reversed the decision and allowed him to publish, the Dutch government, because Dr. Fouchier worked from a Dutch university, required an export license for him to actually publish his paper, because it was under the consideration that he was potentially exporting dangerous knowledge outside of the European Union. Therefore, there needed to be a licensing and review process. Obviously, there are concerns with governments regulating the information that scientists and the research that scientists are able to work and research on. So there was actually a legal conflict between the Fouchier lab and the Dutch government with regards to whether his research actually needed a license. And moving forward, this eventually it was the issue was dropped in court, and he did get the license. But it does pose that conundrum, what extent nations should or regulate synthetic biology research and how the mechanisms by which they are supposed to do so. You also have global governance of synthetic biology. There are much fewer of them, and again, how they're applied by different nations is different. An example they usually come in the form of UN declarations or global agreements. The most prominent example being the Convention on Biological Diversity, which discussed about deliberate release of recombinant microorganisms or organisms, and how the release of those engineered organisms might affect local ecosystems and other non-engineered organisms. So we'll briefly just take a look at some of the major principles that govern how policy and governance is being created in the context of synthetic biology. So the first one is the precautionary principle. This is very much a principle that's put forward by the EU, and essentially it says that if there is a potential and serious and irreversible damage, scientific uncertainty should not postpone the protective measures. So what that means is essentially that if you can surmise that a technology could potentially cause potentially serious damage, even if you're not sure of it, you should treat it as a possibility that needs to be for which measures need to be taken against. So until even if you're not certain about a risk, there need to be mitigating measures present at the time of release in order to make sure that these potential risks do not become uncontrollable. And a lot of this was very much not embraced by everyone outside the EU, just because they were concerned that it might be too restrictive to science. So very much now there is a move, both by EU and the global community at large, towards responsible innovation. So responsible innovation is much more in the idea of anticipating and assessing potential implications and societal expectations before and after a new technology comes onto the market. It's essentially fostering kind of the design of inclusive and sustainable innovation through the consideration of risk and benefits and the development of mechanisms to assess those risks and benefits at every stage of the development. We'll talk about it some more in the next couple of slides, but it's the kind of a cycle of, okay, you start with the technology. What are the concerns that you can anticipate? How is it actually useful to society? How are you going to act upon making sure that this innovation is both useful and does not pose like outsized risks, and how you're going to go about implementing measures and mitigation factors in order to make sure that the risk-benefit balance is maintained before, during, and after your research is completed. There's also the idea of prudent vigilance. That's much more an idea that's been championed or currently used by the US, whereby you establish processes for assessing benefits and risks before and after a project start, but it's very much a watch and wait principle, whereby you allow a technology to come onto the market, and you assess its risks and benefits as it is implemented. It's not so much like the kind of a contrast can be drawn against the precautionary principles, which very much kind of considers all the potential risks at the start and starts taking measures right away before they even become a certainty, as opposed to prudent vigilance, which is more as the technology evolves and becomes available. They think about ways of assessing its risks and benefits and seeing how the technology actually pans out and it can be regulated. So we're just going to talk about a little bit more about responsible innovation, mostly because responsible innovation is the principle that is currently gaining the most traction across different legislations and across different governance bodies. Uh, so one principle, one model for responsible innovation is the area framework. So the area framework involves anticipate, reflect, act, and engage. So what that essentially entails, so first is the anticipate stage, which basically um, talks about what the impact and implication of the technology is, what we expect them to be, what are the intended consequences and the unintended ones, as in what are the ones for which they're designed, and what some of the ones that may happen as a result of the technology being deployed. And this can involve anything from environmental to social and economic consequences. So it's just about thinking what these impacts and implications are. And then you have the reflect stage, which essentially is about talking about the motivation and the purpose of the technology. Does it fulfill the societal need that it is developed for? Is it, you talk a lot about the ethics, but also what are the areas of uncertainty? What are the assumptions that you're making when you are releasing a particular technology? And it's about being reflexive and thinking kind of at every stage, what are the ethical and considerations, what are the social considerations, and is your purpose aligned with those? And then there is the engage stage, which essentially is identifying and communicating with your stakeholders through deliberations, dialogue, and various forms of engagement. And that's very much talking with the people who are involved in developing the technology, as well as people who will be using the technology or will be impacted by it in some manner, shape, or form. And that can be a broad variety of engagement, being engagement on the visions, on what we consider a goal of the research of a type of or field of research to be, with regards to, like, say, funding priorities. Maybe we should fund more research towards one area as opposed to the other because that's something that various stakeholders find desirable or there's a need for it. Impacts with regards to what are the impacts of a certain technology, and what the opinion of various stakeholders are regarding these impacts, and uncertainties. What do we not know about certain technologies, and how do people deal with that uncertainty? Are they comfortable with the level of risk that are being taken with some uncertainties? And that's essentially what we try to gauge at the engage stage. And also act. That's very much the taking actual steps to in your research in order to change your trajectory based on the feedback that you get. So that's through all the previous stages, the anticipate, the reflect, and the engage stage. The act stage is very much where you shift the direction of your research or the implementation of your research based on the information you've gathered. So we'll take a quick example just to illustrate how this area framework works. And this is very much was helped by the framework that was discussed by the 2017 Exeter team from the IGEM Exeter team. So we're using a bit of their framework to kind of illustrate how this area framework can be used for responsible innovation. So say you're designing a bacteria to degrade waste plastic into something more reusable or less pollutive, say a form of pressed plastic that can be more readily used in something else. So if you were to apply the area framework for it at the anticipate stage, you would ask yourself questions like, what are some of the ways this could impact the environment, the economy, the laws, the environmental policies? What are some of the technical challenges you will face? Is there a particular type of plastic that you can degrade, that your bacteria can degrade, but not others? And can all plastics be degraded? How will you contain your bacteria? Will it be in a processing plant, or are you releasing it into the environment, into, like, say, landfills, so that it degrades bacteria to say the plastic to say something more biodegradable? and there's concerns about whether that bacteria is contained or actually released in the environment. Do you need to sort the plastic? Because maybe there's only some types of plastics that actually can be degraded. How polluting are the byproducts? How are polluting other products? So is the thing you're making with your bacteria more or less pollutive than the initial raw materials you started out with, as well as the things that you need to use in order to sustain your biological system. Say you're using a bioreactor. What are some of the inputs and outputs, and what are their economic and environmental implications of using that on a large scale? You also have, how will they be used? How will they be disposed? The different scenarios and visions and milestones that you need to have in your research, in order to, so that what you want to, what your vision is for the implementation of, say, your degradation system, what milestone do you need to reach in order for it to have that desired vision or that desired scenario? And it's about kind of listing those at the anticipate stage, to make sure that you actually hit those. Reflect, you'd be talking about maybe more about, does society need this technology? Maybe there's other alternatives that are more efficient than degrading plastic using bacteria. How are there any ethical consideration, especially say if you're having deliberate release where you're releasing the bacteria into landfills? Can it potentially affect things outside of the landfill? Can it affect other organisms? All these have to go into your your matrix. Are there any uncertainty about the impact of the technology? Are there things that you don't know? For example, is there potential for mutations or for it to act differently with other types of material? Can your technology be misused in any way? Will it undermine any kind of recycling efforts? Say, if people think that you can just have all this plastic in landfills and degrade it using bacteria, will there be less of a concerted effort from people to sort their trash or go for other kind of recycling efforts? And at what stage of the project do we need to reflect? So very much are there points during your research process that you need to kind of stop and kind of take stock of where you're at and where the technology is at and where the technology is going? So say, is it at the start of your research before you start, or once you have the bacteria developed and you start thinking about the implementation process or the scaling up process, essentially putting different points at which we're going to say, we're going to stop here, we're going to start do a reflection on all the different considerations of the ethics and the implementation of our technology, and then after we've done that and we consider it to be good, kind of acceptable or not, we're going to move forward. Also, so now the engage stage, where we were talking about engaging stakeholders. So you have to first of all identify your stakeholders. Who are they? Are they the plastic companies? Are they the people who are using the water system where your bacteria could be released? Are they the workers whose job may be affected by, if like traditional recycling plants are no longer in use because they're now using your new technology? Is it the industries that will benefit from whatever degradation product that your bacteria produces if it has some other use? Is it the governments that will have to be regulating through environmental policies or through industry guidances how your technology is to be used? So you have to really kind of bring all the stakeholders, and you have to consider, who has control, who benefits the most, who is the most affected, and maybe who is anyone underrepresented in this. Generally, say the government and the industry is generally well represented in the discussion, but some groups may not be as well, have as much of a voice, and it's your job as someone who's trying to engage the public in this discussion to make sure that they're equally represented in that engagement process. And then it's about thinking, what's the optimal form of engagement? And we talk about some of it in our previous lecture on public engagement. But there's different ways of engaging people in a discussion, and how you're going about evaluating that, and also how you're going about integrating the feedback that you get from your engagement activities into the decision-making process that you have with regards to your research and the implementation of it. And finally, you have the ACT stage, which is where when you're talking about essentially how does your research activities and how your goals change based on the considerations you got. Maybe there is a concern, if you're talking about deliberate release, with it spreading to other ecosystems where it may be harmful and mutate. So maybe you have to consider having fail-safe systems in your bacteria so that it only survives in very defined environments, or based on, say, regulated, having kill switches or different dependencies on certain substances or nutrients, so that it does not survive outside of the desired setting. Maybe you, at the level of stakeholders, you need some kind of educational training programs to better prepare the industry for the technology that you're going to be using. Or maybe you need to work with the government to make sure that your technology is properly regulated. All those are ways that you can kind of integrate the feedback that you got from engagement and reflection and anticipation into the decision-making process of implementation and your kind of scaling, as well as your research activities. So we're just going to touch briefly on the general approaches to biotechnology governance. And when we dig into each of the individual countries, you will see those approaches in various forms, but these are the general ones. So you have the first one is the human rights approach, which is basically courts usually take decisions based on any kind of technology that may have some discriminatory or stigmatizing practice. And they put regulations in place so that these can be controlled and regulated, and you prevent any kind of discrimination or stigmatization. They're usually based on constitutional rights that are articulated by courts. So usually this form of governance usually happens after a technology is implemented into and impacts society. The great benefit of governing a technology in this way is that you actually express the public values in considering constitutional rights and kind of preventing any kind of discriminatory practice. You clarify the issue, and you kind of set precedents for how future technologies are to be regulated. The issue with this, though, is, for one, this can only happen once the technology is implemented and it's actually become a product in society. It has less of an impact at a more upstream stage, at the stage of research. It's also time consuming. Legal discussions and legal battles in court take time, and that can both delay the commercialization of certain technologies, as well as maybe allow them to be unregulated for a substantial amount of time while things are being worked out in court. Another limitation as well is that the scope of court rulings are generally very limited, being quite specific to a certain case. Governing sustainability in this way tends to create a number of court decisions that are very much limited to the case for which they are applicable for. You can have statutory specific approach. So that's more in terms of laws that are crafted issue by issue regarding the implementation of different scientific advances. So these come in the form of prohibitions, constraints, moratoria, on how a technology is to be used, what is allowed and not allowed based on a law on a specific technology. The great benefits of that is that it provides immediate certainty, clarification, and precision on how a specific technology is to be implemented and used, and what's allowed and not allowed. It usually shows the political consensus and is the result of deliberation. So it will at least show be a decision from that the government agrees with. But the issue again with this is that it generally has a limited scope. Generally those laws apply only to a set of technologies, and they're not very wide ranging. So you may actually have certain statutes that will contradict each other based on the technology they're applied for. And as new technologies develop, they may fall in a bit of a gray zone between those different laws that have been articulated. And there's also the danger that it may close public debates. Once the government has put a law on something, it may discourage people's ability to debate that technology and having discussions on it, because it's almost like a verdict has been reached and there's a way of regulating it, when there's actually maybe more discussions required. Another approach is the regulatory approach, and that's more of an industry-based form of governance, where the government has a code of conduct or a set of quality assurance or monitoring practices that it has for various technologies, or maybe some kind of licensing process. And the industry has to comply with those approaches or those steps in order to be in line with government approval. And usually that's well integrated with commercial and professional stakeholders, because regulatory approaches are generally discussed quite heavily by the relevant industry as well as the government. So they're usually well integrated at that stage. The issue with a regulatory approach that it doesn't really define the moral societal values underlying those regulations. So if you think of a regulatory approach within the context of, say, medication, this would be, what kind of safety studies need to be done? What kind of efficacy studies need to be available? How do you make sure that a certain facility is adequate for the manufacture of a certain therapeutic? How are you making sure that every batch that you are producing is of the quality required to be within the government regulations? How government inspections go about? All of this is with regards to what the regulatory approach entails. But again, all of this are quite technical and between the professionals and commercial stakeholders in the government, not so much defining the moral societal values that maybe were more present in say court rulings or in the statutory approach. And finally, you have the market-driven approach, which is kind of the more free market way of controlling synthetic biology, which is it has the benefit of allowing the research and innovation to be driven by market forces. So it promotes generally the most research, but there are certain caveats with regards to the interests of private and public funders having a bit of an outsized effect on how decisions are made, given that it is driven by money and the market and the demand and the supply. Some groups who may benefit from it may disproportionately have a say in how the technology is regulated, or maybe there are different market forces applying pressure on it, so there might be a lack of consensus with regards to what kind of technology makes it in the market or not. And also there's obviously it's prone to litigation and consumer choices, which don't always have a kind of a guiding moral framework. It may be more based on the interests and rights of different people that are most concerned with the technology. So no government has a purely regulatory, market-driven or a statutory or court-based approach to synthetic biology or to biotech in general. It's usually a mix of those different approaches, and as we'll see in the coming examples, but they're generally kind of the prevailing approaches to which these technologies are generally regulated. So in the EU, they're the one that generally are known to have so kind of the more stringent regulations on synthetic biology. There are three laws in the EU that govern. There's two laws that are actually sorry that govern synthetic biology in the EU. The 2001/18 EC is regulates kind of deliberate release with regards to GMOs and the trials and cultivations and release of genetically modified plants. Any kind of a process, it's very much a process-based approach of qualifying risk based on what technology was used, depending on what technology was used to create that organism. So is it CRISPR or gene editing, or is it maybe some kind of cross, or is it some kind of plasmid that you're integrating into the genetically modified organism? They're regulated very much, the risk level assigned by the EU on those organisms is based on what process was used to create them, to develop them. You also have the second regulation is the 2009 41 EC regulation, which applies more to microorganisms that are used in continuous, not so much to things that are deliberately released into the environment. And it classifies it more on the risk to health and the environment. And it details the kind of the minimum requirements and measures necessary for containment at each level of risk. So you guys, if you're interested into doing more of a legal assessment of it, you can dig a little bit more into those two laws or directives from the EU with regards to how they're regulated currently. There's also the European Food Safety Authority that governs how things are released into society and the risk to ecosystems. They are more concerned with the environmental impacts than they are with the risk of the actual organism in its intended use. So that covers all the risk to ecosystems of these GMOs and they specify kind of protection goals and assessment endpoints. So what should we strive to protect, and to what extent we need to protect them, and how we're going to go about assessing whether those protection goals are achieved. And that involves kind of defining what acceptable risk is. So what is something that a risk level that is acceptable to the environment, to the ecosystem. The laws defined by the EFSA and the Convention of Biodiversity very much talk about what is acceptable risk, what is protected, and over what time period it is allowable. So let's move on to the EU, to the US. The US generally errs more towards the kind of market-driven approach to things. So, but it still has, as well as the regulatory approach to things. Previously, the RAC was also involved in, so that's the recombinant DNA Advisory Committee, did all of the regulations on synthetic biology. But now it's been moved on to various other organizations like the NIH, for example, who are concerned with not only with funding, but also with kind of research with biosafety, and they apply kind of like the biosafety levels that you're familiar with, kind of biosafety 1 to 4, based on the potential harm for the organism that you're dealing with, any kind of like potentially harmful synthetic DNA segments, as well as any kind of like vectors that you're using. And the way that the NIH regulates it is very much in considering the synthetic segments very much to be regulated the same way as its natural counterpart. So say you were to use something from a pathogenic strain of E. coli, you were to take a biobrick from that, or only a piece of DNA from that, and put it into another organism, the NIH would regulate it in the same way as you kind of like having that pathogenic strain, as it would regulate the pathogenic strain, depending on how it's actually being integrated. Another organization is the EPA, that also mostly concerned with the environmental aspect of things and the environmental release and the effects on the ecosystem. Aspects that are involved with genetically modified plants are usually governed by the U.S. Department of Agriculture's Annual Plant Health Inspection Service, the APHIS, and things to do with food and drugs are under the jurisdiction of the Food and Drug Administration. Very much at least for the FDA, it's very much an adaptive regulatory model that they use, and that they use in terms of regulating it. And as you can see in the US, there are various agencies that deal with various type of synthetic biology technologies, and one of the drawbacks of that is that maybe there is sometimes certain technologies may not neatly fit under the jurisdiction of any one institution, and you may have some confusion into how it's currently being regulated and which institution needs rules and guidances need to be applied to that technology. Just a look at larger in Canada. So in Canada, we have very much a product-based regulatory framework. That means that, say, a new technology is used to design food and pharmaceuticals. We assess it based on what the end product is, not very much by how it is made. So that's very much in contrast with the EU that we discussed previously. The EU was very much concerned about whether technology you use to create it, be it like recombinant techniques, like cell fusion or otherwise, and what heritable material you were transmitting to the organism. But Canada is very much more involved with the end product. What is it that you made, rather than how you made it? And it assesses the drugs and pharmaceuticals, not the process by which it was made. And Health Canada, Environment Canada are the two main regulatory bodies. So food and pharmaceuticals are regulated by the Food and Drugs Act, and environmental risk assessment is performed by the Canadian Environment Protection Act, according to that. And it's very much the regular approach that Canada uses. So now what I wanted to really for you to get out of our discussion so far is that different entities govern synthetic biology differently based on the influence that they're able and how they actually matter as a stakeholder to the research and to the implementation of the technology. And we've looked a little bit at how different countries have gone about regulating synthetic biology, and maybe what are their approaches or their tendencies with regards to what model they're using in terms of regulating those technologies. But the one thing that you will see very much from every major governing body at a state level is that they perform some form of risk assessment on a new technology, basically saying what are the risks of that technology and how we're going to go about putting regulations based on how risky it is. So now we're just going to have a bit more discussion about risk assessment in general, and how you go about doing some kind of risk assessment. So effective risk assessment is based upon having measurable criteria and parameters. These need to be things with limits and defined specifications as to what is something that is considered toxic or dangerous, as opposed to what is considered something as acceptable in other cases. It needs to be a transparent process, so that all of these have to have well-documented guidances that both the public, the industry, and government has access to. There needs to be proportionality of risk. So what that means that your mitigation strategies need to be proportional to the level of risk that your technology is having. So for a low risk item, you maybe will have fewer mitigation requirements as opposed to more of a high risk technology. And effective risk assessment requires consistency in decision making. It's very much about setting a set of rules that are to be applied across all the products that are regulated by a certain risk assessment model. So the key concepts we'll be discussing about is problem formulation, which is very much identifying what the risks are for a particular technology and how you're going to go about assessing them. Hazard identification and characterization. This is more about talking about the actual technology in terms of, how is it as an entity, as a chemical entity or a biological entity, harmful to humans or to the environment, and what kind of characterization do you need to do on it to be able to define the level of hazard that entity, that technology has intrinsically and as in its finished product form. You also have exposure scenarios, which is in what way do people or the environment interacts with that technology. The level of uncertainty. So in any kind of new technology, you will have a level of things that you're not aware of. And say, for example, and we'll take that example later, of whether something may mutate, or whether the biological stability of a microorganism is in different situations. So you have to account for the level of uncertainty in your risk assessment matrix. What's considered acceptable risk? At where do you draw the line between what is a level of risk that you're able to live with within a certain level of mitigation strategy? You need a defined analysis plan, so you need to decide prior to starting your risk assessment how you're going to go about analyzing your research, what your information needs, what information needs to be provided by whoever is applying for the new technology to be assessed, what information they need to provide you in order for you to make a fair assessment, and what are the evaluation criteria. And that's very important to define before you even start into what are kind of your limits, what are your yes or how do you go about making your decision, what are the lines kind of drawn out for you to say yes or no on certain levels of risk. So we're very much using the risk assessment for synthetic biology. We're using the risk assessment model that was proposed by the EU in an opinion that the EU asked to be drafted for Synbio to give you an idea of how this is not currently how it is being regulated, but this is how they're proposing for it to be regulated in the future as technologies evolve. It does actually involve a little bit of how it is currently regulated, but it builds upon it. So there's the precautionary principle that we discussed previously, considering uncertainty as a part of risk assessment, having anything that of the risk that you can potentially have. You consider the mitigation strategies that you need to have in place, even if that risk is not a certainty. Case-by-case risk assessment. So very much the nature and level of detail depends on the living organism modified, and the traits, the use, as well as the potential effects on the environment. So it's very much risk assessment is not a one-size-fits-all. It's a case-by-case thing. Comparative approach. So risk is considered in the context of the risk posed by the other non-modified recipients or the parental organisms and in the potential environment. So you need something to compare. In order to assess something, you need something to compare it against. And the EU recommended that you compare, say, first of all, the risk of your host organism to the non-modified recipient, the parental organisms, the non-modified host. What are the risks against that? And then what are the risks in the potential environments that it is being put into? And it's very much about having some kind of standards for comparison. And then finally, it's a step-by-step approach. So the EU very much recommended that you start from the highest level of containment. You assume that LGM was to be regulated at a very high level, at a very kind of like in-depth and taking the most precaution available, and then gradually you reduce the containment, and as you are able to determine the level of risk through studies. As things are shown to be gradually safer, you can reduce the level of containment or the scale of release. So, say something has initially with a high level of uncertainty, maybe is assessed as requiring more containment. As that technology is shown to be safer over time, maybe you can reduce the level of containment and increase the scale of release. So this is again taken from that opinion from the EU regarding how synthetic biology could be regulated, but it also kind of showcases how a risk assessment kind of decision tree usually is structured. So on one side you have the hazard identification that we discussed previously, which is basically saying, what is the potential harm that this technology may have as itself and in its finished product on humans or the environment. And based on that, we provided a risk class. And the second one is where you're talking about exposure. So what are the different scenarios in which people might be exposed, or the environment might be exposed to, and to what amount they would be exposed in each scenario. And one of the factors that are involved in when we're talking about exposure is the type of manipulation that was done to the organism, because that very much impacts how risky the exposure is. Once you consider the risk characterization and the exposure, then you start assigning a class of risk for, in this case, the continued use of the activity, but it could also be very much for the deliberate release of a technology. That's through a risk assessment bit. Then you get into risk management, which is how you're going to go, what are the steps that you're going to take in order to make sure that the risks that you've identified are properly controlled and not harmful to humans and the environment. And there's very much a cyclical step here about every time that you implement a containment level per se for your continuous, you need to reevaluate your risk class based on that. So if we were to go by the EU recommendations of gradually decreasing your containment levels as things are shown to be safer, say you start out with a very high level of containment, and then over time, as you are able to show that your technology is safe, you may reassess that level of risk. And the end goal obviously is maximum protection of human health and the environment. So that's why you are taking all those risk management steps. It's the ultimate goal is to protect humans' health. So again, just hazard identification. That's a step of identifying what are the possible deleterious effects that you may have on humans, animals, and the environment in terms of toxicity. You talk about allergenicity, pathogenicity. With regards to the environment, maybe you would consider the adverse effects on biodiversity, on how that would affect the ecosystem, how it would interact with other organisms. So that's at the hazard identification step, and you would then go about characterizing those effects. So you would do tests on toxicity, on how on the level of pathogenicity, of transmissibility of the technology, and based on that hazard identification, you would assign a risk class, and a bunch of factors go into that decision process. What is the severity of the disease or infection that you may have as a result of of an engineered microorganism? What's the virulence, the infectivity of a particular strain? What are based on the modes of transmission? How big of a dose do you require for you to be infected, and what are the routes of infection? Also, one consideration with regards to genetically engineered organisms is with what's the host range, what is the specificity of the target. species relationship. So can your microorganism only impact, say, humans or a certain type of animal, or does it have the potential to affect a broader range of hosts? What's the biological stability? Is there potential for your organism to mutate heavily? Or is there for it potential for it to change in some significant way at a biological level and at a DNA level? And also, what's the potential for survival and dissemination in the community or the environment? So is your organism resilient enough to survive outside of, say, a contained environment, or outside of the designated area that you want to release it to? And that's very much going back to kind of biological kill switches and fail safes that we put into that might be required for certain organisms in order to make sure that they're not able to survive outside of its desired environment. And another consideration is the availability and effectiveness of therapeutic measures. Say the organism you're engineering does not have a vaccination for, or an antibiotic, or some kind of therapeutic agent that can protect you against it, then that would pose a higher risk. Or say there is a potential for emergence of a resistant strain. Again, that would put it into a higher risk class. The prevalence of the microorganism in the environment. So is there any kind of like native microorganism that organism could interact with and potentially pass on traits to the native organisms? The presence of target species around the installation or the site of waste disposal. So is there any species that could be impacted by this new organism or technology that you're putting with in around the place where it's being installed and the place where the waste that contains that engineered organism is being disposed, as well as any kind of exotic character of the microorganism? Is there any way that the microorganisms behaves differently from your comparator or from other organisms of its kind? With regards to some of the considerations that you need to have for the type of manipulation, maybe it's in the form of, what's the recipient microorganism? So are you putting these genes into E. coli, or maybe are you putting it into some kind of native microorganism in a certain environment? That's a consideration into what kind of exposure you will actually have. What genetic material is inserted, and from what organism, what's the donor organism? Is it coming from a pathogenic strain? Is it something that could increase infectivity or pathogenicity? What vector is being used to transfer the DNA? What the donor microorganism is, again, what kind of not only what DNA, what you taking from that organism, but also what the risk of the native organism is, as well as the resulting genetically modified organism. Is there, once you've engineered an organism in a certain way, does it now have properties that are different from both the hosts as well as the donor organism? Is there something different in the way it acts, and that can impact the level of risk that you assign to something? And with regards to the probability of exposure, which again is that second branch of our risk assessment tree, it's about talking about the potential for aerosol generation. Can it be carried by air? The scale of the activity. Are we talking about releasing something into the environment at a few mils, or are we talking about huge container sculptures like liters of culture of your recombinant organism? Also, the concentration and volume. To what extent is everyone in that this needs to be considered is exposed to what concentration they're exposed to, what volume they could be exposed in different scenarios, or in the event of a failure of a certain containment, and what type of work is being proposed. So is your technology to be used in vitro? Is it to be used in vivo? Are you working with lab animals? Are you doing any kind of non-standard manipulations? And how have you actually calculated your probability of exposure? So have you done, again, kind of animal studies, or maybe have you done challenge studies where you've tried to use your organism in, say, a human clinical trial to define the probability of exposure and what the impact of that exposure has. So now we talked the last decision tree that we referred to was very much about continuous. This is more with regards to deliberate release. Deliberate release obviously having an environmental dimension to it as well. And again, it's very much, this figures from the synthetic biology opinion piece that was requested by the EU with regards to what are recommendations on how synthetic biology is to be regulated. So with regards to, again, we still have the same steps of problem formulation, which is identifying the hazard and what the potential risks are. And that breaks down into your hazard characterization, doing tests to see the extent of that hazard within the organism and any finished product that may be created as a result of it. And the other branch is the exposure, is now that you know how hazardous you've done two tests like, say, cancerogenicity, teratogenicity, and other kinds of toxicity assays. Now you have to talk about to what extent people will be exposed, or the environment will be exposed, and to what concentration they will be exposed at. Based on those two factors, you characterize the risk, and you decide on a risk class and the risk management strategies that you need to have in place in order to mitigate that risk. And then you can come up with an overall risk evaluation and conclusion. So specifically talking about deliberate release, we are going to talk about some of the factors that are involved in that. So one is persistence or invasiveness. So that's how long can the organism sustain itself in the environment that you're releasing it in. To what extent can it infect other organisms or other entities. Gene transfer, the potential for it to transfer any kind of genes from itself to other native organisms or to other species. The interaction with the target, as well as the interaction with non-target organisms. So that's maybe, say, in the medical context. Say with regards to, say, we're talking in the case of gene drives, and the potential for gene drives to, in the context of malaria and controlling the spread of malaria by putting a gene drive in certain mosquitoes, so they spread the genes that prevent reproduction of potentially contagious mosquitoes that could spread the disease. And in this case, you talk about the interaction of that gene drive with, is it only mosquitoes that to whom the genes could be passed on, or could it be passed on to any kind of other organism? The impact on cultivation, management, and harvesting. So what are the impacts on agriculture? Any biogeochemical processes that may be impacted by the deliberate release of any natural or man-made geological or chemical processes or cycles that can be affected by it. And generally just human and animal health. How, if an organism, a genetically engineered organism, is released into the environment, what are the impacts on the health of humans and animals that could happen. We're just going to end with some of the recommendations that that opinion piece actually had with regards to how Synbio was to be regulated, and that really kind of works into what work you could be doing in terms of your risk assessment and furthering synthetic biology. So there's very much a discussion that they require further characterization of biological parts. So as we discuss, when risk assessment is done, you need a comparator. And as you further characterize different biobricks and determine their safety levels, you are able to establish the safety of various comparators. And this way you do not have to every time kind of go back to the original natural organism in order to have a comparator. Say your system is made of 3 biobricks. If you have characterized experimentally or using predictive models what the risk of each of those biobricks are, you then only have to, you can kind of make assumptions about their safety, then you only have to characterize what their risk is as a whole and not necessarily as individuals. And then there is a requirement, they recommended standardization of the submissions of genetic modification data and genetic parts information to risk assessors. So very much at the moment there is not a standard way in which all this information is provided. Streamlining that process would very much make it easier for regulators and risk assessors to make decisions on different technologies. The use of GMOs with a proven safety record as acceptable comparators. Again, that's with creating be it hosts or vectors that are very much the standard for which we know the safety profile for those. And that would help in the assessment and having a comparator. Developing risk assessment methods in parallel with Synbio advances. So that's very much with, as the technology evolves, risk assessment method needs to also evolve in parallel so we can keep up with new technologies. And information sharing about specific parts, devices, and systems is very much about the transparency aspect of risk assessment, which requires that information be available so that people can have confidence in how and understand and prepare themselves for how technologies are assessed in different scenarios. So I know this was kind of long and maybe a bit technical with regards to risk assessment, but this is very much what we wanted to talk about with regards to governance. Again, this lecture is very much just an intro into all those topics. If at any point you do want to dig into any one of those aspects of governance at every level and with different approaches and in different legislations and countries, there's very much room for you to look into that and how where risk assessment sits right now and how it can be further improved upon is very much an active area of research that has quite some promise and actually does need to be in place for new technologies that are going to come on the market. I would very much recommend that you take a look at Dr. Deborah Matthews' course at Johns Hopkins on engineering lives in bioethics and public policy. They were very much the basis of a lot of the governance discussion that we had in this lecture, as well as the various opinion pieces that we discuss from a risk assessment base that was made by the EU Scientific Committee on Health and Environmental Risk, as well as the Exeter team from IGEM in 2017 made a very interesting framework on how responsible innovation is to be applied. So I would very much recommend that you take a look at that. And yes, thank you for your attention. And if you have any further questions, please let me know.